Welcome, everyone, to today's Explainer. We are unpacking a genuinely fascinating paper today that fundamentally shifts how we look at a really devastating condition. You know, when we think of Huntington's disease, we almost always picture protein aggregation, right? We think of these toxic clumps of mutant protein just building up inside neurons. But today, we're taking a completely different angle. We're going to look at the cell's internal highway system, because the central message of the paper we're examining is that Huntington's disease isn't merely about protein clumps. It is literally a massive cellular traffic jam right at the Golgi apparatus.
Okay, let's dive into this. Here is our roadmap for today's scientific journey. We'll start with the normal pathway, then look at the key players, HTT, OPTN, and RAB8. After that, we'll introduce the disease variable, locate the actual traffic jam, look at the resulting lysosomal failure, and finally wrap it up with the fully integrated story.
All right, Section 1, the normal delivery pathway.
To understand the problem, we first need to understand how this system is supposed to work. Think of the lysosome as the cell's acidic recycling center. It's a harsh environment, and it desperately needs worker enzymes, like Cathepsin D, to break down proteins, lipids, and just general cellular trash. So let's track a healthy delivery of this enzyme. It starts out as an immature precursor in the rough ER, where it folds up. From there, it travels to the Golgi apparatus, the cell's main sorting center. Here is where it gets a vital molecular shipping label, mannose 6-phosphate, or M6P. Once tagged, a specific M6P receptor, or MPR, grabs onto it at a region called the trans-Golgi network. Next, an adapter complex known as AP1 and a coat protein called clathrin bundle this cargo into a tiny membrane carrier. Once that carrier travels to and fuses with an acidic endosome, the cargo is dropped off, the receptor recycles back to the Golgi, and Cathepsin D finally matures inside the lysosome so it can do its job.
Moving on to Section 2. Let's meet the key machinery making this trip possible. HTT, OPTN, and RAB8.
So, forming a neat little carrier at the Golgi is great, but it isn't enough. It's kind of like having a fully loaded shipping container sitting at a dock. It still needs delivery trucks and motors to actually leave the port. First up in our trucking fleet is RAB8. This is a small GT piece, which is basically just a molecular switch. It's not the motor itself, but it acts as the active signal. When it binds to a molecule called GTP, it turns on. It acts like a beacon on the carrier, signaling, hey, we are ready for transport, and it begins recruiting the next essential players to the membrane. One of those crucial effectors recruited by an active RAB8 is optineurin, or OPTN for short. Optineurin is an adapter protein. Really, you can just think of it as a molecular connector. It literally bridges the RAB8 signal on the carrier directly to the motors that do the heavy lifting, specifically motor proteins like myosin VI, which physically generate movement by walking along actin filaments in the cell. And finally, we have normal Huntington, or HTT. Now, obviously, it's famous for its role in Huntington's disease, but normal HTT is actually a massive scaffold protein. It acts as a dynamic spatial organizer. Its job is to ensure that all these moving parts, the RAB8 beacons and the OptN connectors, meet at the exact right place and time to stabilize this whole trafficking complex at the Golgi. And this brilliantly illustrates how HTT stabilizes this whole system as a dual platform. Notice the two distinct tracks here. You have an actin-associated side driven by RAB8, OPTN, and myosin V. And then you have a microtubule-associated track driven by HTT, HAP1, and dynein or kinesin motors. This two-sided organization is absolutely crucial. It's what allows these carriers to efficiently connect with multiple motors and physically tear away from the trans-Golgi network. Which brings us to Section 3, the disease variable. Let's see what happens when things go wrong. As you might know, Huntington's is caused by a genetic mutation, specifically an expanded CAG repeat in the HTT gene that encodes the amino acid glutamine. To study this, the researchers used immortalized mouse striatal cells. Now, why striatal cells specifically? Well, because striatal neurons are highly vulnerable, and they are the ones heavily impacted in human Huntington's patients. The researchers compared healthy 7q cells, which have a short, normal polyglutamine tract, against mutant 111q cells. Those 111q cells contain the toxic expanded mutant HTT, and this setup allows us to see exactly how the mutant protein alters cellular function.
Section 4. Locating the traffic jam. We're going to walk through the evidence in figures 1 through 8.
Let's step into the scientist's shoes for a minute and look at the exact experimental evidence. The first question they asked was, is mutant HTT even in the right place? To find out, they used immunofluorescence, basically using glowing antibodies to visually locate specific proteins under a microscope, and they compared the HTT location against a known Golgi marker. They backed this visual data up with Golgi fractionation, which is where they physically spin the cells down in a centrifuge to isolate and measure just the Golgi membranes themselves. The initial findings here are honestly striking. Mutant HTT and our key connector protein, optn, haven't just disappeared from the cell entirely. Their total overall levels are completely normal. But they are severely reduced specifically at the Golgi. They have been displaced from the exact location where they are desperately needed to organize that transport platform we just talked about.
Okay, so they're displaced, but the scientists needed to establish causality. Is the mutant HTT actually causing this optn displacement? The answer is a resounding yes. When the researchers forced normal cells to express mutant HTT, optn immediately left the Golgi. When they took mutant cells and put normal HTT back into them, it rescued the optn localization. And if they knocked down normal HTT using siRNA in healthy cells, those normal cells developed the exact same displacement defect. So the crucial point is this: normal HTT is absolutely causally responsible for keeping optn anchored at the Golgi.
Next up, they used immunoprecipitation. This is a neat technique where you essentially pull specific protein complexes out of a cell to see who is holding hands with whom. They paired this with a specific Rab8 mutant called Q67L. This is a version of Rab8 that is artificially locked in its active on state. They wanted to see what happens when that molecular switch is jammed and can't flip off.
And the data here reveals something super interesting. Rab8 and optn can actually still physically bind to each other in the disease model. However, much less HTT joins this complex. The overall scaffolding is just structurally weak. Furthermore, when they locked that Rab8 switch so it couldn't turn off, it stalled the entire transport process, perfectly mimicking the mutant HTT defect. So abnormal Rab8 cycling and mutant HTT both end up disrupting the exact same crucial step.
So what does this broken scaffold mean for the actual physical cargo trying to leave? To see this, they used high-resolution electron microscopy to look at the tiny structures, alongside a really cool technique called IFRAP. Essentially, they laser bleach the cell's fluorescence, but leave the Golgi glowing. This gives them a starting line, allowing them to start a stopwatch and see exactly how fast cargo manages to escape the Golgi.
Now, what's really interesting about this slide is the undeniable visual proof of the traffic jam. The electron microscopy showed that the AP1 and clathrin-coated carriers are successfully forming. They look totally fine, but they just dangerously accumulate right next to the Golgi. They literally can't leave. And the IFRAP timing proved that cargo like LAMP1 and our Cathepsin D receptor exit dramatically slower in the mutant cells. The shipping containers are fully packed, but the trucks have no motors to pull them away.
Which takes us to Section 5, lysosomal failure and accumulation. This covers figures 9 and 10. If all this vital cargo is stuck at the Golgi, you have to wonder, what happens to the poor lysosomes downstream? To measure this, researchers used LysoTracker, a dye that lights up acidic compartments.
alongside a brilliant tool called DQ Collagen IV. Now, DQ Collagen IV is awesome because it only glows when it is actively being chopped up by proteases. So it allows researchers to measure real live degradative activity happening inside the cell.
The downstream effects are frankly tragic for the cell. With those NPR receptors stuck at the Golgi, the lysosomes simply do not get their mature cathepsin D. Instead, the cell just fills up with the useless immature form of the enzyme. Because of this, live degradation absolutely plummets, which they proved by watching the DQ collagen IV fluorescence drop. Without functioning recycling centers, cellular trash just piles up, leading to a massive, toxic accumulation of autophagic vacuoles, which they visualized using MDC staining.
Section 6. The final integrated story.
Let's bring this all together and see how this builds into one cohesive mechanical model. It all starts with mutant HTT failing to properly associate with the Golgi. Because that vital scaffold is gone, OPTN and RAB8 get displaced, and the motor platform is effectively torn apart. Without those motors to provide pulling force, fully formed clathrin-coated carriers just accumulate at the trans-Golgi network. Because the cargo goes nowhere, Cathepsin D delivery plummets. Ultimately, the lysosomes starve, they lose their ability to degrade waste, and toxic cellular trash builds up, eventually driving neuronal death.
Which leaves us with a huge, critical realization. For decades, the primary focus in Huntington's research has been on clearing out those protein aggregates. But if this disease fundamentally involves a catastrophic Golgi traffic jam, how must future therapies pivot? Could we design drugs to somehow re-anchor optineurin, or maybe stimulate RAB8 to get these carriers moving again? It is a fascinating paradigm shift that opens up entirely new therapeutic doors. Thank you so much for exploring this incredible cellular journey with me today. Keep asking questions, and I'll see you in our next explainer.